Pacinian corpuscle. So what are these Pacinian corpuscles? The Pacinian corpuscles are these pressure receptors. And this is an example we need to know in A-level biology. So the Pacinian corpuscles, which are pressure receptors, and these are located beneath the skin, and also other various internal organs as well. So each of these Pacinian receptors are connected. So these are connected to a sensory neuron. So each of them are connected to a sensory neuron. So if any stimulus has been detected, or in this case this receptor detects pressure, it can be detected really quickly. So these detects pressure, and within the membrane of the neuron there are these special thing called stretch mediated sodium, let's make that neater, channels. So they produce the stretch mediated sodium channels, and when these channels change shape or stretch, the permeability to the sodium changes. So the permeability, when there is pressure applied, the permeability to Na ions changes. So if we have a look here, the Pacinian corpuscle. The Pacinian corpuscles are pressure receptors. They are located in the skin and also in various internal organs. Each is connected to a sensory neuron. Within the membrane of the neuron, there are special stretch-mediated sodium channels. When these channels change shape or stretch, the permeability of the sodium channel changes. So here we have a normal Pacinian corpuscle, the nerve ending here, but when pressure is applied, there's distortion taking place here. You can see they're located beneath the skin and also the internal organs as well. So if we have a look here, the Pacinian corpuscle here, a diagram that shows the different effects. We have the Pacinian corpuscle, pressure is applied, distortion of the nerve ending here, and therefore when there's distortion, an action potential takes place. So when pressure here, so when pressure therefore is applied, an action potential takes place here, and therefore the response is taken down to the sensory neuron. What else can we know? So we know that within the membrane, the neurons in the neuron, there are special stretch mediated sodium channels. So when these channels change shape, when they stretch, their permeability to the sodium channels also therefore changes. So if we have a look here at this here in more detail, the stretching opens the sodium channels and creates a generated potential, and this leads to an action potential in the neuron. So when there's pressure applied here, as you can see in this direction, it changes the distortion, changes the charge on this bit, and therefore generated potential. This leads to an action potential in the neuron. So this then causes that to become positive as well. And in more detail here, the Pacinian corpuscles change the mechanical pressure into nerve impulses. So in its normal state, the resting state, the stretch-mediated sodium ions channels are too narrow to allow the sodium ions to pass through them. So the without pressure, it's too narrow at the resting state. When the pressure is applied, the Pacinian corpuscles, it changes shape, and the neuron surrounding its neuron is stretched. So when the membrane stretches, the sodium ions channels widen, allowing more sodium ions to diffuse into the neuron. In influx of the position, sodium ions changes the potential of the membrane. It becomes depolarized. This results in a generated potential. So when influx of positive sodium ions changes the potential of the membrane, it becomes depolarized. This means that this results or means a generated potential. The greater the potential, creates an action potential, a nerve impulse that passes along a sensory neuron. The action potential will then be transmitted along a neuron to the central nervous system, as we can see here. So we have, in a normal state, the resting state, the stretching media of the sodium channels are too narrow. They're so narrow so that the sodium ions here, because you can see they are ions, they cannot pass through these sodium channels because they're too narrow. But when pressure is applied to the Pacinian corpuscle, the membrane changes shape and the membrane surrounding its neuron is therefore stretched. So when the membrane stretches, the sodium ions channels widen. The sodium ions can now go through as the sodium ion channels has been widened, allowing the sodium ions to diffuse in. The influx of the sodium channels diffusing in, the positive sodium channels becomes depolarized. This results in a generator potential, as we can see here. And this creates a generator potential which leads to action potential in the neuron. So this is transmitted along to the central nervous system. So that is about everything about the Pacinian corpuscle.